Good morning, everybody. Welcome to this webinar from KTN and Hamamatsu on photonics for medical diagnostics. My name's Matt Wasley. I'm the photonics lead for the KTN, and I'm joined today by Peter Seitz, who's senior technologist at Hamamatsu Europe and adjunct professor of optoelectronics at EPFL in Switzerland. By Katjana Ehrlich, who's a researcher on the Proteus Interdisciplinary Research Project based at the University of Edinburgh. By Richard Harvey, who's group leader for medical and analytical at Hamamatsu Photonics UK. So you'll be hearing from all of us during the day. What we're going to do is we'll have a bit of time for, or hopefully, depending on the agenda, a bit of timing, a bit of time for Q&A after each talk. But we've allowed time for Q&A at the end. And the way that will work, you've all got a Q&A box on your Zoom screen there. So if you type questions in that, and we can take them as we go along, and the speakers, if they can answer them, they feel free to answer them, and then we'll take them at the end. And we'll aim to finish by 11:45 because I'm sure everyone wants to stretch their legs and get a comfort break at that point. So do remember that we've got Meeting Mojo networking set up for this. So if it was a physical event, which we sort of missed to greater and lesser degrees, we'd all have a chance to sort of meet during the coffee and whatever. So we can't quite reproduce that, but this is a really good opportunity for you to sort of meet the speakers and the other attendees. So we've got a huge amount of people registered for this, so thank you very much all for coming. And people are still joining us as we're talking. So I think to keep us on track, that's probably enough from me. So I'd like to start us off with Professor Dr. Peter Seitz. So, Peter can take control and start us off.
Thank you, Matt. So as soon as I have control, I will start. Good morning, everybody. I'd like to start with a picture that is surely very familiar to all of you, in particular during these taxing times. I do not yet have control over the slides. Now, here we are. So, this is a question surely most of you have asked yourself. You don't feel so well and you don't know what's the problem. What's wrong with you? The dream would be that you have a lab always with you. For example, a lab on the keyring. So the only thing you'd have to do is put some body fluid on it. For example, you spit on it, or you sweat on it, or perhaps you put a blood drop on it. And then this lab on the keyring tells you exactly what might be wrong with you. All of you know we don't have that yet. This lab on the keyring is still a vision. What is missing? What disruptive or known technology is there that could turn this vision into reality? Well, perhaps it's photonics. And I'd like to share with you some breaking news from the European technology platform Photonics 21. As you see, they helped to develop a saliva test with a miniaturized lab to detect a COVID-19 with lasers. And if you read the text, then you see that what they are claiming is ultra-sensitive laser biosensor. Yes, this works. Is it new? No. Actually, the technology is quite old, 30, 40 years old. Okay. Is there other technology that could come to the rescue? Well, everybody knows about spectroscopy. So is there a spectroscopic solution? Optical, electrical, nuclear magnetic resonance, mass spectroscopy that could help us here. Let me illustrate this with a typical near-infrared spectrogram, in this case of vanillin. So a spectroscopist doesn't have to smell vanilla. He just needs to look at the near infrared and the mid-infrared spectroscopic recording, and then he can tell what you should smell. By the way, this is a very interesting spectral range from 3 to 10, 12 microns, and it will surely play an important role in the future, but not in helping to solve our problem. Let me explain why. So the difficulty is in this, I admit, very complex diagram. But it's complex because our body is so complex. Did you know that we are expressing about 100,000 different proteins? This is what your body is manufacturing. And I'd like to point out that the dynamic range of these different analytes in the blood is huge. So you see here, that's 12 orders of magnitude. And that's the real problem. So we need to find individual molecules in a large, very complex soup. So in order to present you with a take-home message that is easy to remember, let me illustrate the point of what is so difficult in medical diagnostics and biosensing with a picture book you may know. So Waldo will help me explain, illustrate what is so difficult in biosensing. Now, you all know the picture. So somewhere here is Waldo. Where is Waldo? So you see, our biosensing problem is very much the same as finding Waldo. We need to be very sensitive. We need to pick out Waldo in this mess of objects and people. Now, that's the first problem. We need to be very sensitive. The second one is illustrated by, hey, I see something. Okay, so the arrow does not appear. It should have pointed to this object here. If you look closely, then you will see, oh, yes, it's a person. Yes, it looks similar to Waldo, but it isn't. So this is Wenda. Wenda looks similar, but she is not Waldo. So that's the second problem. We need to be very selective. We need to be sure that it's exactly the object, the person we need to identify. The next problem is very often it's not one object, one analyte we need to detect. It's several. So it's the thief, it's the sorcerer, and many others. So in practice, biosensing means measuring, detecting a panel of objects. So we would like to simultaneously detect different analytes. So that's our next problem, this simultaneous measurement. And a last problem, and this is now not a problem of your screen or the transmission, this is something I introduced purposefully. So this is a picture that is not stable. So if it's not stable, you'll have a hard time finding the object. So that's our fourth demand. Whenever we would like to realize a biosensing solution, it must be stable. Okay, and now you understand what I mean if I say that we have a 4S challenge when we do medical diagnostics or biosensing. we want sensitivity, we want selectivity, we want simultaneous measurements, and we want stable measurements. We need a reference that is completely flat and reliable. So that's it. What are the physical implementation mechanisms that we would have? So here they are. These are all biosensing measurement techniques that I know, and I'm sure you'll recognize your favorite one in here. As you see, I've made 4 distinctions, namely optical and non-optical methods, and I have labeled and unlabeled methods. I'll explain in a second what I mean with labeled or non-labeled. It turns out that the most sensitive method is a combination. It's a combination of chemistry for the actual transduction of concentration of molecules into another signal, and then using an optical method with which to read out this transduced signal. So that's the best you can do today. I won't have the time to explain why this is the case, so I'm just going on to my next slide, telling you that even if you know it should be an optical measurement technique, there are many different types you could use. So you could measure a change in refractive index, a change in the amplitude, a change in the frequency, or a change in the luminescent properties. So these are the possibilities you have. Most of the time, and in most of the products you find today, it's the refractive index change. In a way, the simplest you have that is used. So now that we know that, I'm able to show you how we can measure optically in labeled and unlabeled, label-free conditions, the analyte concentration. So in blue, I have the sample fluid. In a sample fluid, there are swimming a lot of molecules. In particular, we have the analyte. So that's the target. That's what we'd like to know. We have many, many other molecules in large concentration differences, and we use a receptor. So this is the simplest way of using receptors. We just have a sauce, a soup where the receptors are, and then if you put the analytes in, the receptor will bind to the analyte, and this chemical reaction will make a change in the optical properties, actually the refractive index. So that's the simplest solution, and nobody uses it. People use, for example, this technology. They bond, they affix the receptors on a surface, on a substrate, so that a flow, a microfluidic flow of molecules, analytes and other molecules flowing by will have a chance that the receptors capture the analyte. So we don't need to shine light everywhere. It's just the surface that we need to inspect. What you see here are label-free optical surface biosensing. And I can already tell you that's one of the most often used technologies in optical biosensing today. You could also use reporter molecules, fluorescent molecules that are bound to the analyte. This has the advantage that we have a much increased selectivity, because we need the selectivity that the analyte binds to the antibody, and this reporter, this luminescent molecule, binds to the analytes. So we have the product of the probabilities that we actually know that it's this analyte and nothing else. It's the target we want to measure. So we can excite again with light the fluorescent molecules, and then we just count how much light is emitted. And a final possibility is very similar. The only thing that we change is first we let the liquid, the sample, flow over the surface so that the analytes can bind to the receptors. And then we let the fluorescent molecules flow over the surface so that they bind to the analyte as well. Now for the terms. This column is called label-free optical biosensing. This column is called labeled optical biosensing. And the easier ones, as you can imagine, is label-free. You don't need any additional fluorescent molecules. The advantage of this second line, where you have receptors that are bound on the surface, it's possible that you also find out how fast does such a binding occur, which is very important if you want to find a new drug. So from now on, I'm concentrating on the lower left corner, label-free optical biosensing. So what we need to do is we need to team up different technological solutions to make a concentration measurement. First of all, we have a chemical transduction. This gives us the selectivity. We can measure how much did the optical properties change once this binding occurred. And we don't have to look into the complete volume of the fluid. It's enough to look at the surface. In a second, I'll tell you what this means, surface. Then we need an optical readout mechanism. The sensing is preferentially done with an interferometer. So what you see here is actually in one arm of the interferometer. And if you see any change in the resulting light coming out, then you know, this is due to a change in the effective refractive index here. The advantage of such a solution is it can be really small. I'm going to show you a practical example later on. And you'll see, due to this smallness, the temperature, which influences a change in refractive index tremendously, is the same in both arms. So you have high stability as well. So such a miniaturized system, that's exactly what we wanted. Remember this keychain thing? It could be done because a typical size of these interferometers is a few square millimeters only. Today, you find products that use one of two surface sensing technologies. Both are employing evanescent waves. These are propagating waves, propagating in a dielectric waveguide, but there is also some decay into both of these parts here, in particular into the sampling fluid. This can be done with a dielectric slab waveguide, but it could also be done by gold or silver on a dielectric substrate, which is shown here. This technique is called surface plasmon resonance, SPR. And to the left, we have grating coupled waveguides. You immediately see that this decay distance is of the order of a few tens to a few hundred nanometer. That's exactly what we need. Here now, a symbolic illustration of how the technological solution looks like. In the case of grating coupled waveguides, we have a grating that couples in light of the appropriate wavelength, and then this light can be transmitted in the waveguide. In the case of surface plasmon resonance, we typically use such a prism. Now, all of you are very clever, and I'm sure that several of you thought, aha, if I need to sense the refractive index here, and I have light that can propagate in this waveguide, why don't I put a mirror somewhere so that the light is going back and forth, so we can extend the sensing distance? By the way, this wouldn't work for this technique here, because metals absorb radiation, and therefore after a few hundred microns only, light has decayed too much. A resonator wouldn't work here, but a resonator would work here. Hey, do we have the solution we've been looking for? Very, very sensitive? Unfortunately not. Let me show you. Here is a picture of a range of optical micro-resonators. It is true, they are highly sensitive. The more resonance, the higher the Q factor, the higher the sensitivity to sensing a change in refractive index. This is true. But if you implement a sensor according to this label-free optical technique, you'll find this surprising curve. This is how sensitive is the detection mechanism to the presence of analyte molecules. And you see there is a sweet spot. If you go too high by the Q factor, somehow you get less sensitive. I need to explain. But the important point for you is you should be in this sweet spot where you have resonant mirrors, where you have grating couplers, where you have planar waveguides or surface plasmon resonance. So you see what is being done is done because we are at this sweet spot. But why does it exist, this sweet spot? Well, because of the geometry of chemistry. Let me explain. So if you have a resonator, the higher the Q factor, therefore the better the resonator, the more confined is the light to the center. So the higher the resonance quality factor is, the less light we have in this volume here where the biochemistry takes place. And let me remind you of the geometry of the chemistry. Here it is. So you see here, typical antibodies, they have a size of, let's say, 10 to 30 nanometers. And what we have to measure is this effect, namely the analyte combines with the antibody, and we need to measure here 15, 20 nanometers above the surface whether the refractive index has changed. So if you concentrate all light here below where the action takes place, you won't see a result that is related to the change in concentration of analytes. So that's the real problem we have. Two pictures here to bring the message finally home. A typical protein measuring many 10,000 Daltons. One Dalton is the mass of one hydrogen molecule. So this tells you more or less how many atoms are involved. So we have here 10, 20, 30 nanometers. The glue we are using to glue the antibodies onto the surface is typically streptavidin. That's not so big. So it's really the proteins that are behind this effect. So now you may be convinced the solution is here. I just told you that we can measure things on a few square millimeters with the required sensitivity, and we're done. Let me show you the highest sensitivity product currently on the market by the Swiss company Creoptics. This is the machine doing these measurements. So the sample, the liquid sample, is put on sample test chip that is put into this drawer here. Let me show you in more detail. So this is the drawer, this is the sample, and the optics, all of this interferometric thing, it's the same magnification here, is happening here. 1-2 square millimeters. But what is the rest good for, this machine? And the answer is, this is for the microfluidics, this is for the refrigeration, this is for the computing, this is for keeping mechanical stability. In principle, we are there. Yes, we can do very small miniaturized optical biosensing. The problem is all the other issues, they take so much volume, so much space, and the largest part of the price. Is there perhaps a solution on the horizon that it could become cheaper? Not really. Nevertheless, let me share with you the most recent development is this in this domain, published by Roche Pharmaceuticals, and it's called molography. So they take this combination of reference channel and measurement channel to the extreme. They don't have just two of these channels, one reference, one measurement. They combine many in parallel. It's a hologram. And the hologram is made such that the high ridges are covered, are sensitized, the lower ridges are unsensitized. So you have a miniaturized chip that is very insensitive to temperature variations, very insensitive to non-specific binding. So that's the future. I'm at the end of my presentation. Let me quickly summarize what I told you. So medical diagnostics is not easy. The number of analytes is huge, many 10,000. The concentration range is huge, 12 orders of magnitude. Therefore, the biosensing solution must meet the 4S criteria: sensitivity, selectivity, stability, and we need to make simultaneous measurement for an analyte panel. The most successful biosensing solutions on the market combine chemical transduction and optical sensing. In particular, interferometric changes, measurement of phase changes. State of the art is either grating coupled waveguides or surface plasmon resonance. Perhaps the future is molography. But my most important message comes last. Mastering photons is not enough. The big machine, the big volume and the price does not go into photonics, does not go into chemistry. It goes into all the problems of microfluidics, chemical surface functionalization, stabilization, keep the things stable also mechanically, and solve the stability problems outside the photonics. With that, I'm at the end of my presentation, and I hope Waldo did his job well. You remember what it means to be excellent in biosensing and medical diagnostics.
Thank you very much, Peter. That was really impressive sort of walk through the the state of play and what's happening. Also, thanks very much for joining us from Switzerland, which just shows the power of being able to run an online event. We're going to whip through because we're a bit pushed for time, but I'll, as is usual, I'll take one burning question, and we've had a few in the Q&A, and I'll take in no particular order. And so everyone's put a question in the Q&A, so we'll pick as many as we can up in the Q&A at the end of the session today, and then we'll produce a Q&A document if there's some big burning questions. So I will take one burning question. I'm just going to pick one at random. So James May asks, where do you see the role of AI and machine learning techniques in this type of sensing?
I don't think that AI plays an important role here, because the hard task, namely selectivity, is done by chemistry. If you want, AI could play a role in finding the right receptors. And as far as I know, there is some work being done in optimizing receptors guided by AI. So it's the chemistry, but definitely not the photonics that could profit from AI.
Okay, thank you very much, Peter. We've got questions sort of mounting up, so we'll have plenty to talk about in the Q&A at the end, I have no doubt. So thanks once again. I'd like to move on now, and I'd like to ask Dr. Katjana Ehrlich to take over here and tell us about the great things going on in the Proteus project.
Yeah, thank you for the introduction. I currently have not yet control. No, I have control over the slides. So there's a lot of delay. Okay, thank you for the introduction and for the invitation. My name is Katjana Ehrlich and I'm a postdoc at the Queen's Medical Research Institute at the University of Edinburgh. First, I would like to highlight that the work I'm going to present here is a huge team effort, and I'm presenting the work of a lot of my colleagues. So if you want more information on a specific topic or application, afterwards, please contact me and I will put you in contact with the respective person. So going to my... So I will first give an intro. Okay. First, giving an introduction to the Proteus project, the motivation and the basic technologies we're using before I go into the applications, optical molecular imaging, fluorescence lifetime imaging, and fiber-based sensing in detail. So the challenges to the healthcare systems are numerous at the moment and cannot be solved by one research discipline, one team, or one institute alone anymore. We need fundamental research, clinical, translational science, and industrial partnership to make an impact and to make a truly translatable device. This all is present at the QMRI and the Edinburgh BioQuarter. Proteus itself is one of the 3 interdisciplinary research collaborations funded from the EPSRC, and it combines researchers from 3 universities, University of Bath, Heriot-Watt University, and the University of Edinburgh, with several collaborators, industrial partners, and academic partners such as Durham or Dundee University have joined over the years. Each of the universities bring in a different discipline to the team. University of Bath, optical fiber design and manufacturing, Heriot-Watt, photonic device manufacturing through ultrafast laser inscription and optical system design. Edinburgh University brings in chemistry, biology, medicine, and the clinical environment due to its closeness to the Royal Infirmary and the NHS. And all researchers work at their individual universities, but are also co-located at the QMRI and our so-called hub, where everything comes together, and this is really the strength of this project. The motivation from the Proteus project comes from the clinical drive. So lung diseases are numerous, and the current diagnosis methods do not give clinicians the information they need for fast and accurate diagnosis. So clinicians are facing a diagnosis dilemma. They have structural information from imaging modality, but these are often not specific enough. However, taking a biopsy or lavage sample for, okay, this remote access is a bit tricky. However, taking the sample from a biopsy or lavage and performing a histopathology, accurate diagnosis is possible, but it takes often days for the results to come back and allow stratification and targeted treatment of the patient, and valuable time is lost. So Proteus aims to combine fiber-based in vivo imaging and sensing with histopathology through optical molecular probes deep inside the distal lung. Focusing on patient and critical care, which are ventilated, we are aiming to develop a low-cost fiber-based platform based on disposable optical fibers that can reach far into the lung and advanced detector technology combined with bespoke optical molecular probes. If we visualize the whole system needed for a fiber-based in vivo imaging and sensing system, we see that researchers from several disciplines are needed. Their skills and their experience are needed to work together as close as possible to build a working device and overcome the challenges of the way to clinical translation. So the bespoke optical fibers we use in our project are designed and manufactured by the team of the University of Basel. A few examples of fibers we developed for the project so far are high resolution and low cost imaging fiber bundle, which also minimizes the effect of core-to-core coupling, a holocore fiber for background-free Raman sensing, and a multi-core fiber for multiplex sensing. Here we also see a finished device, which is ready for endoscopic application. It is comprised of 3 fibers, an imaging fiber bundle, and 2 capillary fibers for microlavage and smart probe delivery. The imaging, these 3 fibers are packaged in a biocompatible tubing, and the total device has a diameter of 1.4 millimeter, which is compatible with the most working channels of standard bronchoscope. For optical molecular imaging, a multicolor wide field fluorescence endoscopy platform was developed, which is based on LEDs, off-the-shelf optical components, and commercial CMOS cameras. The current costs are 5,000 for a single build, but you can imagine they can be easily lowered for a larger scale build. A wide field approach was favored instead of a confocal approach to keep the costs low. However, it comes with the drawback that extensive imaging processing is needed in order to achieve the same resolution as the current clinical confocal system. It has been shown that when the lung tissue is excited with blue light, it fluoresces in the green. Dominantly, we see the fluorescence from the elastin structure, which you can see here in the top image. This is used by clinicians for guidance, but also allows the differentiation between healthy tissue and disrupted tissue, which you can see in the lower image here. A disrupted tissue structure that indicates a disease process, such as cancer and inflammation or scarring. The device has now been deployed in clinical studies, and I will show some results later. The advanced approach is a confocal laser scanning system, which we are currently developing. This system is based on pulse lasers and galvos for scanning, and is therefore a more extensive system, but also a more expensive system. The system already has a good imaging quality, optical resolution and contrast, and it also features the fluorescence lifetime imaging through optical fiber for endoscopic application, and it's currently on its way into a clinical study. The time-resolving technology we're applying in this project is based on single photon detectors developed by the CMOS sensor and system group by Professor Robert Henderson of the University of Edinburgh. This detector has 512 pixels and are based on 130 micrometer CMOS technology. It features 2 different spot types, which are sensitive for different spectral regions. One is more sensitive for shorter wavelengths and one is more sensitive for longer wavelengths. And it has 3 readout modalities: single photon counting, time-correlated single photon counting, and on-chip histogramming. And especially the last one is the most exciting one and will enable real-time time-resolved imaging in the lung. Optical molecular probes, or smart probes as they are sometimes called in literature, consist of 3 parts: the recognition element, which is specifically made to attach itself to its target, which can be bacteria or an enzyme or any other key marker for a disease or cancer. Then a linker, a dye, or fluorophore, which changes its emission properties due to changes in the molecular structure or its environment. If the recognition element comes into contact with the fluorophore, comes into contact with the molecule, the fluorophore lights up. And we have a library of these optical molecular probes developed in Proteus, and this is one of our one that is currently on its way into clinic. It's BAC2. It's specific to gram-negative bacteria, and you can see here an image with lung tissue, autofluorescence in the green, and on top labeled bacteria. which you can see as little dots on top of the lung structure. And you can imagine in a video that would be like a twinkling you see or a blinking. So coming now to the applications. As I mentioned before, our wide field fluorescence imaging system, Versicolor, is currently employed in a clinical trial where we try to visualize activated neutrophils and cancer. The activated neutrophils are labeled with an optical molecular probe that fluoresces in the green, whereas the CMED, a hallmark for cancer, is labeled with a smart probe that fluoresces in the red. And unfortunately, the videos do not work here, so you have to like imagine these are videos. You can see a lung structure, but what we see here in the middle is the baseline imaging of the autofluorescence from the lung tissue, and what you see here on the right is the lung structure overlaid with fluorescent signal from the activated neutrophil, from the labeled activated neutrophils, and the labeled marker for cancer in the red. So, however, one problem we discovered when using optical molecular probes which light up in the green, and several fluorophores approved for in-human use are fluoresce in the green. It is hard to distinguish between the heterogeneous structure from the lung tissue or cells and the tiny labeled bacteria which appear either as dots, as you can see here, or twinkling during imaging in a moving and breathing patient. This is what we call the green-on-green imaging problem, and it can be tackled in a lot of ways. One option is to use a fluorophore that fluoresces in a different spectral region, for example, red, as shown in the previous slide. We can also use data algorithm that can find and track these blinking dots. Two other options we've been working on in Proteus is on the photonics side, and I would like to present them here. So it's spectral ratio endoscopy and fluorescence lifetime imaging. Ratio endoscopy uses differences in the spectra of tissue and bacteria for contrast enhancement. What we see here are the spectrum of alveolar tissue in red and the spectrum of labeled bacteria in black. And we see that they are different. So we can now separate two channels. We have the channel in green here, which is the lower wavelengths, and a channel in red, which consists of the longer wavelengths. And if we separate them and take the ratio of the two of them, we can clearly distinguish between bacteria, labeled bacteria, and the lung tissue. You can see here on the right side some standard fluorescence intensity images of lung with labeled bacteria, and it's very hard to distinguish if labeled bacteria are really present. However, if we take the ratiometric approach here, we can clearly see that labeled bacteria are present because they have a different ratiometric values, and they are lighting up here as yellow areas on top of the violet lung structure ratiometric values. Another option is fluorescence lifetime imaging. The fluorescence decay rate, or the fluorescence lifetime, which is the average time that it takes for a fluorophore to relax after excitation. On the left side, we see an image of lung tissue and labeled bacteria through a standard confocal microscope, and we can get spatial information and fluorescence intensity information. If we now take the same image through fluorescence lifetime imaging, we can clearly distinguish between the shorter lifetime of the lung tissue, indicated in green here, and the longer lifetime of labeled bacteria in red here. So the confocal scanning system will have the capability to do fluorescence lifetime imaging, and we can see here first images from the system. We have of a convallaria slide. We have here fluorescence intensity image and a fluorescence lifetime image on the right side. And this is the first image of lung tissue on a microscopy slide stacked together through the system. The system is currently being further developed and for a clinical study, which will start soon. So far, the fiber itself was only used as a passive sensor, and we also want to explore point sensing in the lung. We do not only want to image, but we also want to explore point sensing in the lung, which was additional to the imaging application by integrating another fiber into the final fiber device. Here we have prepared labeled bacteria onto lung tissue and performed time-resolved fluorescent sensing through an optical fiber. You can see here the fluorescence decay after excitation. The decay combines the fluorescent signal from lung tissue and labeled bacteria. We can see clearly that there is an increase in fluorescence lifetime when labeled bacteria are present compared to the pure decay from the lung tissue, which is here in the black. If we take now liquid from the tissue bacteria sample, kind of performing a lavage, the difference becomes more significant. And it's also shown a difference between labeled live bacteria and dead bacteria. We do not currently know what is the exact process and why we are seeing the difference between live and dead bacteria when they're labeled with the same probe, but we know that the morphology of the live and dead bacteria is very different. So far, the fiber itself was used as a passive sensor, but we also want to sense physiological parameters localized in the lung. And we transformed the distal end of the fiber into a sensor for this. For fluorescence sensing, we functionalized silica microspheres, displayed here. The microspheres are loaded with two fluorophores, carboxyfluorescein and TAMRA, for pH sensing. While carboxyfluorescein is pH sensitive, TAMRA is not, and the ratio between the two signals, which are spectrally different, can be used for pH sensing and has been used before as well. The sensor is a multi-core fiber with the cores etched back so that we have holes where the loaded silica microsphere fitted tightly in. They fit really tightly in, so you just push them in and then they stay in. You can rub on your hand or you can rub on paper, they just stay in. So there's not, but they might be packaged a bit further for really clinical use. But what we've done with the sensors so far, or my colleagues, is using it in an ex vivo lung perfusion model. We have access to transplantation lungs that are not deemed fit for transplantation. And in one of our studies, the Innovate study, we test the physiological parameters of these lungs and evaluate why they are not fit for transplantation, or if they are actually would have been fit for transplantation. But after these tests have been carried out, we can use it for our devices and test them in there. And the ratiometric pH and oxygen sensor has been tested in this ex vivo lung perfusion model, and a good agreement between the pH and oxygen sensor and commercial probes were reported. Another point sensing technique for pH in the lung is Raman, we explored in our project. Raman is inherently a very weak signal, which is further overlaid, especially in vivo application, by the Raman signal from the optical fiber and the autofluorescence from the tissue. So we combine here time-gated Raman spectroscopy with surface-enhanced Raman spectroscopy to overcome the limitations. The first part is the fiber sensor itself, here visible on the right side. Our fiber probe consists of a standard off-the-shelf telecommunication multi-mode fiber. On the distal end of the fiber, we deposited functionalized gold nanoshells, which enhance this Raman signal significantly. These nanoshells are functionalized with formicaptobenzoic acid, which is known to be a pH-sensitive molecule. Two of the vibrational bands are affected by the environmental changes in pH. And you can use the intensity of these two bands to measure the pH. These nanoshells are deposited on the end of the fiber and coated with a sol gel layer to not get lost during measurements. The time-resolving spectroscopy, we are able to separate most of the background signal from the wanted Raman signal from the source. The time-resolved spectroscopy, the light travels through the optical fiber, it interacts with the core material, as can be seen here, and generates Raman photons all the way along. which reaches the distal, when it reaches the distal end of the fiber, it generates the wanted Raman signal from the functionalized nanoshells. If we look at the time development of the measurement, we have first information about the dark counts of the detector, then the fiber Raman appears, and at a later point, and only visible shortly, is the source signal we actually want. Having a time resolved measurement, as shown here, which is an actual measurement from this system, it allows us to single out the interesting region of the time on the time axis, and are able to enhance the visibility of the source Raman instantaneously. You can see here, if you use a standard intensity steady state spectrometer of the shelf, you measure something like the red line, the red spectrum here, which is the fiber Raman and as well as the signal from the former captive benzoic acid. There's no autofluorescence of the tissue currently present in this figure. But you can imagine this would overlay it further. If we now apply the time-gated spectroscopy, we can recover an almost background-free spectrum of the former captive benzoic acid signal, which we can then use for pH sensing. So the work I presented here is an achievement of a big team and a huge team effort of the whole Proteus team, as mentioned before. And I would like to acknowledge all of them, but there are more than 50 people, so I will just show you a photo of most of them. And of course, I want to thank the funders and partners and collaborators. And yeah, thank you for your listening. And if you have any questions or want further information, I'm happy to bring you in contact with my colleagues.
Thank you very much. That was really interesting to see what's going on, sort of the wide range of activity that's going on the Proteus project, and good to see the mini submarine as well. So again, in usual way, I'll take the first burning question that I come across, and we'll take some other later. Kejang asked, was the FLIM result in vivo or in vitro?
The FLIM result I showed here is still in vitro, but we have it's not yet in vivo. We have kind of stopped everything in the lab at some point. So these are still in vitro measurements of lung tissue on a microscope slide. But yeah, it's taken forward now.
Okay, thank you. We'll move on, and we'll pick some others up at the end. You can see there's some questions piling up, and I've got a few myself. So thank you very much. I'd now like to hand over to Richard, who's going to talk us through some photonics technologies. Thank you, Richard.
Thanks, Matt. Good morning, everybody. So thanks for the introductions. I'm Richard Harvey, and I'm the group leader for the medical and analytical group for Hamamatsu in the UK. I'm going to give a presentation giving you an overview of some of the areas that Hamamatsu are involved in the current sort of in medical diagnostics, and then just give you some background to the company as well, and just highlight a few products that might look of interest to you. So let's see if I can move the slide. Okay, so there we go. Right. So Hamamatsu was founded in 1953, and it's based in Hamamatsu City in Japan. We're currently sitting at around over 5,000 employees, and in Europe, that's probably over 300. Most of or nearly all production and R&D is carried out in Japan itself, in Hamamatsu City. And we have a huge range of products and devices split into sort of discrete products and systems. So over 15,000 different products. So we won't talk about all of those today, but I'll touch on a few that are particularly of particular interest. In terms of the geographical spread, then, as I mentioned, Hamamatsu's production and R&D is headquartered in Japan. In Europe, then we have main offices in the UK, Germany, France, and Italy, and then some various satellite offices. And obviously, then of course, we have a large presence in the US and in Asia as well. So the company itself is split into several divisions. And these divisions they have strengths in certain areas of different types of technology. So The solid state division, as the name suggests, is primarily focused on solid state devices and semiconductor materials. So this is anything with silicon, indium gallium arsenide, indium arsenide, antinomide, and we'll touch on a few of these products momentarily. The electron tube division then is probably the sort of the founding part of the business in the 50s, and it started off making television pickup tubes. Today, then, one of the core technologies is the photomultiplier tube. So any kind of vacuum-based technology is a real strength area here, but also light sources as well and X-ray sources. The systems division, as the name suggests, makes systems. So this is taking these discrete components and building it up into larger, more end-user focused products. We'll touch on some of these, but something, a typical example would be a scientific level camera. Laser promotion division. They make lasers, quantum cascade lasers being one of the most interesting, and also there's a real strength in lasers for range finding. So we won't be talking about that today, but LiDAR is obviously a hot topic, and that's an area that they're working on. And I'll just show you at the end just one slide regarding the central research laboratory. So in terms of the application spread that Hamamatsu is involved in, I mean, this slide doesn't really go into enough detail at all. But in terms of sort of the medical diagnostic side of things, then blood analysis is clearly a key area, especially in today's in the situation we face today. Hamamatsu has a long heritage of supplying detectors into this application space. Another area of real strength is in medical imaging. So PET and CT and SPECT gamma cameras. A really strong area of Hamamatsu supplying sensors into this area. Other areas then include PCR and high throughput screening for drug detection or for drug creation, flow cytometry, and obviously then there's a large strength in digital radiography and computer radiography, particularly in the dental market. Okay, so I've broken the presentation down into products based on the kind of the division that they're coming from the company. So the first section will be focused on semiconductor devices. This seems to be kind of the best way to give a general overview of what we're doing today. So from the solid state division, then there's a lot of detectors or general photometry, silicon photodiodes, silicon avalanche photodiodes. Detectors for visible, UV, and near infrared and infrared as well. What we'll focus on today is more devices for spectroscopy. So you can see at the bottom we have spectrometers and some devices for low light detection called silicon photomultipliers. So in terms of the spectroscopy, one of the key areas that we're seeing a lot of interest for at the moment is in Raman and in particularly in surface enhanced Raman. So Hamamatsu probably 5 to 6 years ago released our first consumable SIRS substrate, which is based on a slide format with a small SIRS chip in the center. This has now been taken in, basically built into a complete integrated solution. So this spectrometer here is an example of one of these devices. So this it's a complete spectrometer. It includes a 785 nanometer laser diode, and it fits perfectly in with the SIRS substrate. So this allows label-free optical probing, but also there are a range of fiber-coupled versions of this spectrometer, and as in the last presentation we saw, then this can be integrated into endoscopy probes, so we can do Raman in the endoscopy environment. So this is a real strength area at the moment of interest. Staying in spectrometry, then maybe these have been seen before by some of you who are aware of Hamamatsu. We have a sort of a long heritage now of offering ultra compact micro spectrometers. So the image on the left is the first generation, which was released probably 10 years ago. The next image, which is in the metal can. So this is the world's smallest grating based optical spectrometer. So this is covering 340 to 850 nanometers. And then the next one across is actually a near infrared version covering 640 to 1050. But you can see that basically these are really, really compact, so they fit on the end of your little finger, ranging from the lightest at 0.3 grams. These are finding a real strength interest in the point of care diagnostics market. Anything which is looking at portable handheld instrumentation is a real area of growth for these products, and in particular PCR as well is an area that they're thriving in. And this just gives image just gives you an idea of how the optical setup works inside. So the spectrometer is partly part of the manufacturing process, is a MEMS process. So the image sensor here is looking down the screen, and the slit of the spectrometer is etched into the image sensor itself. So the light passes through the image sensor, reflects off this blaze grating, and then reflects back up onto the image sensor itself. So the whole optical path is effectively wrapped around itself. And this is what allows the spectrometer to be such a compact size. Okay, so if we move from visible up into the infrared, and this product's relatively hot off the press. It was released in the last few months. It's our new Michelson interferometer-based Fourier transform interferometer. This is covering 1.1 to 2.5 microns with an excellent resolution of 5.7 nanometers for something which is basically palm-sized. Now, this device actually integrates a VCSEL laser into the package. So this is giving you really high wavelength accuracy and really high repeatability. So this feeds back into the 4S's of Peter's presentation. This product again comes with full evaluation software and full DLL library, so it's really aimed at supporting our customers into using this instrument as quickly as possible. So moving away from spectroscopy and into low light detection. So this is called a silicon photomultiplier. So some of you may be aware of this. We also call it MPPC, which is multi-pixel photon counter, but we use the two terms interchangeably. It's effectively a solid state photomultiplier. So it has really high photon detection efficiency, excellent timing resolution, and a low dark count. So what that means is it's really ideal for fluorescence and bioluminescence measurements. In terms of the applications that it's typically being used in today, then point of care, PCR, and LAMP as well, areas where it can be used. We're offering this in a wide range of different package options. So you can see on the right hand side, we have two discrete detectors. This type of tiled array here, this is what's really critical for PET and SPECT imaging. And one of the key advantages of this technology is that the MPPC or the SIPM is insensitive to magnetic fields. So this allows customers then to integrate PET with MRI. So now you have these multimodality systems, which we're seeing more and more in the field now. Okay, so if we move on to the electron tube division. So I mentioned this is the division where kind of Hamamatsu started making TV pickup tubes. Now the core business or the core product is in photomultiplier tubes. So this is in the top right. But also from this division are coming various lamps. So typically UV lamps, either xenon or mercury xenon lamps, and deuterium for excitation. X-ray sources. So typically the X-ray sources are used in industrial sector, but actually for preclinical imaging in the CT, these are finding a lot of traction. And there's various detectors for mass spec in terms of microchannel plates. And then these different colored squares are actually scintillator. So different types of scintillator material that we can offer. And scintillator is a material that converts X-ray to light. So if you couple this with a CCD or a CMOS sensor, then you can start doing digital radiography, for example. Okay, so kind of maybe it should have shown this slide before the slide on the silicon photomultipliers, but this is the technology which is the first sort of gold standard for low light detection. So this is the photomultiplier tube. These are really ideally suited for the particular application we're looking at today would be immunoassay analyzers. But actually, they're also still ideally suited for point of care as well. So it's really the advantage is that we have the detector for ideally suited to the application. So there's always a trade-off looking at the silicon alternative to this. Historically, then the key application area for this would be in medical imaging again, so PET, SPECT, and gamma cameras. But we see more and more interest now in point of care as well, and sort of lab-based instrumentation, which can take sometimes the slightly larger size of the detector. So these are available as discrete PMTs, as you see here, but then there's also a huge range of modules and various accessories, including high voltage power supplies, which we can supply as well. So this is still a very relevant technology today. Okay, so mass spec is another real strong area. And in terms of medical diagnostics, then LCMS and GCMS and also MALDI-TOF are regularly used in the diagnostic field. In that area, then Hamamatsu offer microchannel plates. So these are effectively multiplying ions, electron multipliers, and then various ionization sources as well. But we also have a new material or a new product called butane, which is, I'm told that's how it's pronounced today, which is really is a simplified sample preparation technique for moulding. And this image here, so this gives you an idea. So it looks like a slide-based substrate. The advantage of butane over the typical mouldy matrix would be that there's effectively no matrix background noise in the image or in the signal. It has very high reproducibility, so again that feeds back into the 4 S's. There's no sample pre-treatment required, and it has, if you're doing imaging mass spec, then it has very high spatial resolution due to the nanometer scale structure on the chip. So this is something, if mass spec is something that any of you today are interested in, then I'd highly recommend we can speak about this later on. We can put you in touch with the relevant contacts in the UK. Okay, so the final division I wanted to touch on was the systems division. So as you can see from the pictures here, then it kind of gives you an idea that these are more complete products, more end user based products, which you typically see in a lab. And that generally holds true through what we offer. The sort of the core product range here is really cameras. So we're very pleased to say today that we have the market-leading scientific CMOS camera, which is a backed-in CMOS or backed-in SCMOS camera. And I highly recommend you go visit our website to look at the specs on that. It's a really fantastic camera. But alongside that, then we also have a range of CCD, EMCCD, and InGaAs cameras. InGaAs is for your infrared imaging, and we can offer these in both board level and board level and end user cameras. And the board level cameras are effectively PCB based cameras, which are mounted or can be mounted into an end customer's piece of instrumentation. Any type of imaging application, be it fluorescence, time result fluorescence, bioluminescence imaging, again PCR as well is a strength area. These cameras, the high sensitivity and incredibly low read noise make these really suitable for the medical diagnostic market. So alongside this, then the nanozoomer. So this is more of a complete end user system, and it's actually a slide scanning system. So this is basically taking pathology slides and scanning them with a high resolution camera to give you high resolution digital images. The advantages of this type of system are that it gives you the ability to copy and share a large number of files between pathologists, and it also obviously allows digital slide storage rather than physical slide storage. There's a whole range of systems in this product range, but high throughput scanning is one of the key advantages as well. So these can be, the slides can be stacked up overnight and left to scan. There's various imaging options, including fluorescence and the Z-stack imaging, which I think is really cool because basically you image the slide, and you can then image the slide at various focal depths through the slide. So you can effectively zoom through the slide at different depths, which is really nice. And the key thing is that this is an open platform. Now, there's obviously a lot of talk in the news at the moment about lateral flow tests as well. Now, Hamamatsu don't make a lateral flow reader in the sense that it's a medical device. This is not a medical approved device, but it is a tool for R&D and QC immunochromatographic reagents. That's a difficult one to say. So this device then has absorption and fluorescence modes, and really it's aimed at anyone who's making these tests can use this as a QC instrument. So the system on the left is sort of more like the end user version, and then we can also customize this for various needs, adding printers or USB interface, changing the form factor as well. So it's definitely worth talking about if you're on the chemical, on the reagent, the biology side of things, and you need something to give you a real firm answer on what you're seeing. So please do get in touch if you'd like to talk about this further. Okay, so drawing to the end now. Everything we've been speaking about so far has been sort of detection or diagnostic based. This is actually a drug screening system. So this is just a small portion of the overall system, but we call it the FDSS, or the Functional Drug Screening System, and it's basically designed for cell-based assays in the drug discovery field. Now, I'm not an expert on this product myself, so some of these terms are kind of new to me. But I highly recommend, if you are involved in this area, then do get in touch, or we can arrange a separate meeting through the Meeting Mojo and I can put you in touch with our key contacts in the UK who can talk through this product with you in more detail. Okay, so I mentioned the 3 different divisions, and we've looked at some key technologies on key products coming from those divisions. And there was this 4th one, which is the Central Research Laboratory. So Hamamatsu in general then is investing or reinvesting 10% of turnover back into R&D on a year-on-year basis. And the Central Research Lab itself is doing a lot of blue sky research into things like optical materials, energy, and obviously for today, health and medical care. So some of the highlights of recent research they've conducted has been in, for example, the creation of a new PET probe for the study of mitochondrial function. There's a lot of work in near-infrared spectroscopy, in particular for optical mammography. And also then finally some really nice, exciting new 3D imaging flow cytometry systems for imaging circulating tumor cells, or CTCs. Now, this group is always publishing papers, so I highly recommend if you can track them down in the usual sources and read through some of the work we've been doing. And of course, there's information on the website. If you want to talk in more detail, we can do so. Okay, so just to sort of wrap up towards the end. So why Hamamatsu? So Hamamatsu, we consider ourselves to be the world leader in photonics. We have a huge range of solutions from discrete components on the detection and the emission side to complete end user systems. Sort of Hamamatsu really prides itself on its reputation for quality and reliability. And in this medical diagnostic field, this is critically important, particularly for them, as Peter was saying earlier on about being like stability, but not only that, but we're in the stability of supply as well. So our medical-based customers really rely on us for that. We have a huge range of application knowledge based on the huge range of applications we're involved in today. So we can always put you in touch with the relevant contacts within Hamamatsu to discuss your project. And really, we're looking to be a partner. So it's very much looking at long-term relationships and starting from the ground up. So we're interested in talking to anybody at any stage of their business, be that large OEM or a small startup just coming out from a university. So we're open to discussions with everybody. Okay, so hopefully that gave you just a very brief summary of some of the areas that we're working in, some of the key products that we're getting a lot of interest in today, based on the world sort of global climate that we're facing today, and it hopefully gives you a flavour of what we can do. So please do encourage you to sort of reach out and get in touch. So again, I'm Richard Harvey, so I'm the group leader for the medical and analytical. I'd like to say a special thanks to Nick, who helped me with putting these slides together at a very short notice. So many thanks to him. And yeah, so welcome for any questions if anyone has any, or we can speak later on.
Great, thanks very much, Richard. Really interesting sort of overview of the wide range of things there. I'll just take one question now, just pick one off the top of the list, and then I'll go on. So Thomas Kraus asks, and hello Thomas, so he's talking about PMTs and avalanche detectors have got a much higher noise floor than pin diodes, and he wonders why you see them as advantageous, given that time resolution is not an issue for point of care devices.
We're seeing in terms of the PMTs and particularly so avalanche diodes is one thing, and the silicon PM, which is basically an array of avalanche diodes, they're really coming into play when reagent quantity is really low. So it's when we're looking at fluorescence and very, very low light levels, where basically the pin diode, so the raw sensitivity is just not there enough, and you need that additional gain from either the APD or from the PMT. Okay.
Thanks very much, Richard. And we can pick up any more. And I would just encourage anybody that had a question that's just occurred to get it in the Q&A box, because we'll just have time to go through a few. So the presenters have been pretty good at answering all the questions as we've been going along. So if there's any more, please do share them for later. But thanks very much, Richard.
Thank you.
Okay. And it's now me. Oh. Sorry, my screen's just done something a bit strange at the moment. Sorry. Bear with me. Everybody, it was all fine until this bit. Natasha, could you take back control and put me on the first slide, please? Thank you. If I take it back. Okay, if I can get control back. Okay, right. Sorry about that, everybody. Okay, so I'm going to talk you through some of the funding that's available for photonics. I'm going to talk a lot about COVID-specific funding, but we'll also look at some other types of funding for photonics. And probably the first thing I really want to say is it's not an exhaustive list by any means. So all I'm really doing is giving you some indication of the kind of funding that's out there and that you can look for. So places to look to start with. So KTN produces a roundup of all the COVID-specific funding, and that's refreshed regularly. So it's more than I could sort of get in any sort of static presentation. So you get the slides so you can get all the links for these. So we have a specific funding roundup on COVID. We also have, excuse me, yeah, we also have a funding on enabling and emerging technology funding, which would include photonics. And again, that's updated regularly, and it's got lots of different things on it there. Innovate UK, you can go straight to their page and search for funding there, and that'll have all their competitions on. Another useful place to look into going beyond grant funding, the British Business Bank's got quite a helpful finance hub section. So let's have a look at some of the COVID-related funding specifically. So one of the main ones out there is the UKRI Open Call. So it's UK Research and Innovation. And they're looking for proposals. So I guess breaking COVID funding down, you've got the actual sort of treatment of it, and then you've got the impacts of it. And so photonics probably comes in a bit in both, but certainly sort of addressing the impacts to society. And this call is about mitigating the impacts, certainly. And projects of 18-month duration, and it's kind of wide open. And if you go and follow the link, you know, there's lots and lots of scope in there. So it's interesting because it's across all UKRI. So academic institutions can get funding, as can companies that would be eligible to receive funding from Innovate UK. If you've got an existing grant as a university or an existing Innovate project, it is possible to get that sort of change direction and use this scheme for that. The other interesting thing about this is that there is usually you talk about how much budget is available for this, and with this there's no fixed budget. They're interested in proposal that can achieve an impact basically in the timescale. So it's a sort of method of flagging up good research ideas and technology ideas. Innovate UK are doing a number of sort of measures that are supporting the response to COVID. One of them that's had huge amounts of interest lately is the fast track. So businesses supporting or reducing, helping the response to global disruption. And so there was a call which is closed now. It was a de minimis call, which is a bit different from Innovate, and it ran on a slightly different different track. And it was an open call. So there was a huge amount of interest in that. So, and in fact, they released the funding from that in two different tranches. We are anticipating another competition coming along these lines. I don't have the details yet, and I would expect it to come out very soon. So So please do keep checking back, and there's a bit later on about signing up for communications, and we'll let you know as soon as that's available. Based on the first competitions that were out, so it was a 4-question application rather than the more standard Innovate 10 questions, but actually the 10 questions kind of mapped onto the 4 questions, so it was a shorter thing. Projects had to start quickly and be complete quickly, and there's a 50k maximum, so so definitely keep a lookout for that one. For companies that are currently running Innovate UK projects, there are continuity grants, and please note the deadline on that is this Friday. So if that's relevant to you, you need to look at that immediately. And there's also continuity loans, and they're running till the end of the year or until the money's used up. So look at those equally. So they're for basically companies and organisations that are running Innovate projects that are at risk of abandoning the project due to financial pressures they've got as a result of COVID-19. And you'll see there 25 to 250k available, and the loans on a similar thing, but a much sort of larger budget. There's a link there for the grants. If you are running an Innovate UK project at the moment, you've got a monitoring officer, so talk to your monitoring officer about the loans particularly. The recently launched is the Future Fund, managed by the British Business Bank. So now this is an investor-led application. So this is targeting companies that have raised some investment, and it's kind of boosting that investment to sort of help them to grow and help sort of kickstart investment into companies. So it will match 100% of the amount that the investor is putting in, up to a maximum of 5 million. There's information there, so if you are a company watching this, or if you're an investor watching this, you should certainly be aware of that. But if you're a company that's raising investment, talk to your investors about this. So Horizon 2020, and it's worth mentioning, I think, at this point that Brexit notwithstanding, we are still eligible. UK, apologies, that is still eligible to compete for Horizon 2020 projects this year, and in fact there's a photonics call which deadline should have passed, but it's just been extended, but I've not really included it in here because it's too close. But so UK organisations are eligible for this call. So it's a new, in fact there's been 5 new calls from the European Commission, and you can find more details about that on KTN's site. But I'm raising this one because it's specific to, it has specific photonics parts to it. So there's the full ID there, and it's 2 to 5 million EU contribution per project. And there's an option in that to get third-party funding, so that will be sort of consortium or organisations that are then going to distribute the funding further out. And there's a 10 million euro slice for that. Deadline is the 11th of June, so fast moving. So it was only launched last week and it's closing soon, so a rapid response. Again, reiterate, you'll get the slides, so you'll get the link, or just email me and I'll send you it directly. But the call link is there, and you can find the details. And there's a briefing recording, and I always say this with Innovate and with with H2020. If you can look at the briefing, it's so much better to actually find out what's going on. So a few more details on the call. So probably the first thing to say, and it was something that kind of struck me when we heard some of the talks earlier, it's how ready some of these solutions are to getting out there. They just need that push to get into use. And so this is what it's about: is to complete and deploy readily available solutions, not to develop brand new things. But I've just taken some words out of the call description here. So sampling, screening, diagnostic, prognostic systems, including advanced photonic solutions, so they're front and center, and then surveillance systems and analytics involving optical biosensors, perhaps, sensors again appearing there, and then protection, including sensors, and sterilization. So I've seen quite a lot on UV sterilization, for example. and ways of aspects of going for that. So that's on the, I guess on the innovation. I just wanted to talk briefly, because I know there's quite a lot of companies in on this, about some of the business support that Innovate and others are offering. So the Enterprise Europe Network in England and Wales is run by Innovate UK, and there's a service basically where EEN are supporting businesses directly. You get EEN advisors working with the business and working in 3 phases. So there's survive, stabilize, and grow, and there's a whole list of things on that slide. So businesses that have been in receipt of Innovate UK and funding and some other funding can basically access that support. There'll be details of all of that we can provide. Oh, and excuse me, it's gone again. I have an itchy mouse, I think. Okay. Right, my mouse is trigger happy, so apologies for that. So I said that was in England. Wales. In Scotland, the companies who go through Business Gateway, if you've got a Scottish Enterprise Account Manager, that's a good place to start off. There's the business support site where you can find information on that in Scotland. And relatively recently launched, Scottish businesses can access funding information support service, which will actually help with sort of horizon scanning funding and actually applying for things, including Innovate and other grants. So work with you can work with Scottish Enterprise through that, and there's an email address there for that. Similar support available in the Highlands and Islands of Scotland there. Contact details there, and just to reiterate, you will get these slides. And in Northern Ireland, the NI Business Info site is a good place to start. And if you have any issues accessing any of the organisations we've talked about here, just let us know, and myself and my KTN colleagues can help you with that. So, okay, I've talked about photonics and some COVID funding and general business support, but what about the rest of it? Well, photonics funding, some of you will be aware that a few years ago, we used to have enabling and emerging calls and regular photonics calls. There have been some EU calls. There's nothing specific out right now that says photonics, but they do tend to come around. We've had Eureka, for example, so they come from some strange places. But really, photonics, for example, underpins a great deal of the UK's Industrial Strategy Challenge Fund topics. So I'm sure a lot of the UK listeners will be familiar with the ISCF. But if we just look at how photonics appears in here, of aging society, we've just seen diagnosis and medicine. We've just had our whole session of photonics related talk on that. AI and data, so we have the audience of the future, so we've got immersive and technology and displays, digital technologies in there. Future of mobility, I think Richard touched briefly on LiDAR, for example, and all kinds of other applications there, and clean growth, which includes agri, which is a sort of big photonics topic. So actually, there are a lot of challenge-based calls that involve photonics, and really the thing is just to sort of keep following those and see where the application lies, and particularly, I'd encourage you to use events like this and other ones to partner with companies operating in those sectors, so you can sort of collaborate most effectively. There is always, of course, the Innovate UK Smart competition, and the great thing about that is that it's open more or less all year round. So I think the deadline for the current one is in about half an hour. But brilliantly, the next one is already on the Innovate UK website and is about to open almost immediately. So Innovate, the Smart competition, is open to innovative projects in any area. It's really focused on sort of transformational really sort of game-changing innovations. It's a very competitive call. I won't hide that. But 100 businesses each call pretty much get funded and do some great things, including plenty of photonics businesses. I've not got a slide on it as well, but it's quite likely we think that the innovation loans will come out, and again that's something to look at, and photonics companies certainly benefited from that. Unique Lasers, for example, benefited from that scheme. And I also want to give a shout out to knowledge transfer partnerships as well. So, there's a lot of change in funding and in governments, and this scheme is probably more like 50 years now. This scheme has run for years, and it's run under various guises because it is so successful. So at its core, you've got a university and a business that are exchanging knowledge, and they exchange that, and the kind of conduit through that knowledge is a recent graduate or postgraduate that's employed to do that. So if you're looking at a university business collaboration, then the knowledge transfer partnership scheme is certainly one to look at. And that's more or less it on my talk there. We'll take some questions obviously in a bit. Please do go on and register. If you go on the KTN site, you can register for your interest. So enabling in emerging technologies is a good one to look at, but also you can register interest in health and transport and etc. And then you'll get sort of latest notice of events like this, competitions, other opportunities. In terms of applying, we've got a good application guide. And you can get it by that link, and lots of other useful information there. So okay, that's a brief tour de force. I'm going to get a better mouse for the next one, but I'll finish there, and we'll take questions. I think I'll wrap all mine up with all of the others. So. Okay, so the panelists have been very good in firing off the answers to all of those questions, and so I just seem to have one live at the moment. As I said, yes, you'll get the funding links. They're in the presentation, and you'll get the presentation. So that's no problem. If I could invite the other speakers to sort of return to the call at this stage. So just to give the opportunity for any sort of final questions. But one of the things that I wanted to ask, and I'll say, Kichana, for example, if you could address. You talked about the all the stuff that's going on in Proteus, and as we've seen with COVID, there's been a need to sort of rapidly get technology out there. So if somebody comes to you with a wheelbarrow full of money and said, let's get it out there, what are the challenges in rapidly accelerating, for example, what Proteus does to get it into action faster than it would do otherwise, do you think?
Well, a huge bag of money obviously always helps in things, and a lot of good connections, I would say. So the problem research projects and academic projects are facing is that there's like this development stage, and then the actually translational stage is often quite difficult and quite expensive, and especially also in like scaling up the developed devices, the fiber sensors, for example, for just sterilization, toxicity testing. This is something that we faced, and it was a huge challenge for an academic project, just being faced by make 300 endoscopy devices, which will then be tested, sent off, tested, and destroyed at the end, which is obviously no academic output for a researcher at this, as well as there's a problem of with just 300 devices or 200 devices you send off for these testings, you're very small scale for these companies, so you often fall in the gap or they don't take you. So it's very lucky that we got into contact with industrial partners, which last companies who put us into contact of their sterile testing facilities, and like provided us the connections. So, but it's still all very, like, bringing something into clinic is a very long and very laborious process, and we are trying to speed things up by taking a lot of functionalities. A lot of people inside Proteus project are having very close relations to, for example, medical physics at the Royal Infirmary. So we have one person there who we work together with, which we take to seminars, so he knows us very well. And we try to get contact with these people very early on, so they not only get the application, the final document from us and be like, approve it. So we take them on very early on to help us and to guide us through the steps. So it will be hopefully a shorter process, and they know us very well and our technology very well.
Okay, thank you.
But Peter, still, with COVID, nothing is fast.
Yeah, sure.
Especially when it comes to in-human use.
Yeah, yes. I'm sure you fielded a question about that getting there. And Peter, did you have anything to add? I know you've been a lot involved in the commercialization of these technologies.
I think we would be misguided if we thought that the problem is photonic. It isn't. The problem most of the time is chemical. And even when the problem is solved, the largest cost incurs once the complete system needs to be validated. So this takes all the time and the money.
Okay, thank you. We've had some. Okay, we're getting a few questions out. This is an interesting one. It kind of relates, I think. So do we think that the key biosensors will definitely be photonics? You know, what about other techniques? And this is Jim Pallo asks this: is it not more likely to see a combination of these in the same biosensor? I'll leave that open for anyone that wants to jump in first.
I think the reason why photonics is so successful is interferometers are so sensitive. So if you want to measure, for example, gravitational waves, the way to do it is use interferometers, because they can measure changes as small as picometers in terms of path differences. So photonics is very simple, low cost, fast, and in terms of refractive index changes, very sensitive. I would like to remind you of one simple fact of microelectronics, because many people ask me, hey, the solution will be lab on CMOS chip. I'm not so sure, because the threshold voltage of a field effect transistor, a CMOS field effect transistor, typically changes by 1.5 millivolts per degree centigrade. But that's huge, because we want to measure microvolts, perhaps even nanovolts. So keeping temperature constant so that the response of CMOS elements, CMOS active microelectronic components are under control seems to be even more difficult in CMOS chips than with photonics. I was asked, would it help if we had more light? Would it help if we had higher quantum efficiency, a wider spectral range in photonic detectors? And I asked the same question to the founder of Cryoptics. That's the company with the most sensitive solution today. Full disclosure, he was my student. So I could ask him the question without appearing too dumb. And he said, oh no, Peter, all photonic problems have been solved. In effect, I have too much light. I have too many photons. That's not the problem at all. I can provide all my parallel channels with sufficient light. This is not the problem as long as I have a stable photodetector. The problems are microfluidic. The problems are stability of chemistry, mechanical setup, but not photonic.
Okay, yeah, thank you. That's interesting. Well, there's one there for you, Kachana. What level of data analytics have you already applied on green imaging issue?
There have been several images, so data analytics applied. I would like to guide you to the papers of Dr. Sohan Said of our group, who applied several types of machine learning to a huge data set of autofluorescence endoscopy clinical data. So that is a huge, so the current clinical system is Cellvisio, which was gathered a large data sets in America, and he analyzed these data sets with several, I think 4 types of machine learning algorithm. I'm not completely sure which one, but I will post the details for the papers in this as well.
Okay.
And as well some algorithm to actually find these dots, and which is very difficult, and especially like very difficult because to compare if your algorithm really works, because the ground truth is always clinicians manually marking each of the frames, and a video has a lot of frames, so it's very difficult to actually prove that your algorithm is really good or better than the clinicians, because it's still a human who, like, sometimes in the evening annotates slides. So yeah.
Okay, great, well, thank you. I'm going to use my privilege as chair to take a question for myself. Keith's asking, essentially it's a question about the threshold for scoring on an Innovate UK competition. And the specific competition you're asking about, Keith, you don't have that data. It's possible to see actually some of the data for previous competitions, but to give you an idea, you really want to be on a lot of these competitions, so they're scoring, I mean the threshold is 70, but you really want to be scoring 85% or better. So on a 10 question Innovate application form, you want to be going through scoring a lot of 9s, hopefully the odd 10, not too many 8s, and definitely not before. And specifically because Innovate really wanted to sort of get things going quickly on the COVID competition, they didn't offer what they usually do is detailed feedback. So this competition was about get it out, get it faster, get the project started quickly. So you won't get that detailed feedback, unfortunately. But there's a lot of advice available out there, and people to talk about writing a good application. So what I think I'd like to do, because we're nearly at time, we've got a few, so we'll take we'll pick up the last few questions offline. But I just wanted to see if any of the panelists had a final sort of closing thought on let's say sort of photonics for medical emergencies, in any order. Peter, do you want to have any final comments?
I can just repeat my final comment from the last slide. Don't think once you've mastered photonics, you're finished. You aren't. That's just the beginning. Solve all chemical problems, stability problems, use problems, in particular microfluidic problems, and then you come from a very small photonic solution to a big machine solving all the other problems. So we are just a small cogwheel in a complicated, large mechanism. And I haven't talked yet about validation and all these things.
Okay, thank you. Kachana, any final comments?
No, I was just typing the information to the answer. So you caught me off guard. No, I just don't think I have much to add to
okay.
It's true. Photonics is you have a lot of ideas. Photonics usually works, but then doing the actually making a device that is mechanical and chemical stable survives sterilization process is not toxic when it comes to contact with tissue, stays on the sensor and not falls off when it rubs inside the lung. These are the really difficult problems. Having then also software, which we often face problem with, is the software that actually then is able to display the information in a manner that it's easy to assess for people who wants to use it, is also very like a difficult thing. So I agree. Photonic is the easy part. Afterwards, it starts to become difficult.
Okay, thank you. And Richard, from your side, you know, when you've got these kind of business partnerships around sort of photonics for medical technologies, so are there any sort of final thoughts on what those key things are that makes that sort of successful
Yeah, I mean, I agree with the other two speakers in terms of, in some sectors, the photonics part is the easy part, but it still can be challenging in some areas. So I just highly recommend people do reach out to us. We have the expertise in-house to really sort of help you with your sort of detection or your imaging problems, and we're sort of just ready to react to that. So photonics is the easy bit, but it's still the fun bit as well. So just please do get in touch.
Yeah, I like that. That's a good final set. Photonics is the fun bit. Yeah, we'll go there. So what we'll do is we'll pick up the final questions offline. We'll take a note. Thanks, Natasha. I'd also like to thank all the speakers today, and I'd also like to thank Natasha, who's been doing a sterling job putting this together and keeping us right here. We're at the close now, so we'll just confirm with all the speakers the slide set that they're happy to send out, and we'll get that out to you, and the link for this recording as soon as we can, and certainly by early next week. So just to remind you all that Meeting Mojo is available for your simulated meetings in the coffee queue. And that starts from 1 o'clock. You can go and book those, so you've got time to stretch your legs. I would recommend you go outside if at all possible during that time and get a break. Yeah, and we'll leave it there. So we are looking at doing some other of these events, very likely around. Oh, and my mind is gone. So yeah, so we're in the next few months. I think we'll do more of these events. So please do let us know in feedback. You know, if you found it useful. There were some technical challenges, of course. So thanks for bearing with us with those. And stay safe, and we'll see you all soon.
Thank you very much.
Thanks very much.
Bye bye.
Bye bye.
Thank you. Bye.